Okay, this lesson is just for honors biology, and it is about dihybrid crosses, which can be very tricky. I even teach this in my AP class. It's in Blackboard, the presentation. It's also as part of your Punnett Squared lesson. I believe it's number 17. But it goes through it pretty quickly. But if you go into your genetics folder, the file is there. You can watch it anytime you want at your pace. But in your packet, it is on page 65 and 66 and 67. In fact, I think part of this actually might be part of your exit ticket for that particular lesson. So here we go. Dihybrid crosses. Once again, Gregor Mendel, father of genetics. Because you can look at more than just one trait at a time. Genetics is actually really complicated. We're starting at the super basic level here. So we know a Punnett square, which was in the last lesson, predicts the genotypes that came from a particular genetic cross. We look at the genotypes and the phenotypes. Mono hybrid crosses. Mono is one, right? One pair of traits is studied. But what do we do in a dihybrid cross? Well, di means two. Two pairs of traits are studied. Homozygous, of course, means two of the same alleles, and heterozygous means two different alleles. And so we're going to do actually one of the most complicated dihybrid crosses there is so that you can do the easier ones a lot more simple. You have a video clip you can watch on dihybrid cross. You can always go back and click on the link in Power School as well. Not Power School, Blackboard. So what do we do? Let's look at the genotype of the parents. You have a pea plant that's homozygous tall and homozygous yellow. So big T, and big Y, that's its genotype. When they're put together like that, you always keep the T's together and you keep the Y's together, always remembering to write the dominant of each pair first. Now we have a homozygous short plant and a homozygous green plant. Little t, little y. So there's our two genotypes based off the given phenotypes. So let's look at think about all the possibilities. And if you remember from math class, the FOIL method, first, outer, inner, last. I like to FOIL it. Unfortunately, I can't write on the screen with you. But there are going to be 4 different gametes for each parent. And so because parent 1 is big T, big Y, let's do the FOIL method. The first, big T, big Y. The outers, big T, big Y. The inners, big T, big Y. And the lasts, big T, big Y. Now, in this particular case, because they're homozygous for both traits, they're going to be exactly the same. But I still want to write it out like this so you can see the method. Parent 2 is recessive for both traits. So first is little t, little y. And the outers, little t, little y. And the inners, little t, little y. And the lasts, little t, little y. So now we have 4 different possibilities of gametes. Now you get a 16 square Punnett square. So you put the gametes from the first parent across the top and the gametes from the second parent down the side. And what if this sperm fertilized this egg? Or what if this was a sperm and these eggs? It doesn't matter. But the offspring, what's it going to look like? Well, now we recombine our 2 letters or alleles for tall, and we combine alleles for the color. So we're going to have a tall plant with yellow seeds, but it's going to be heterozygous. And we do the next one, which will also be big T, big Y, little y, and the next one, and the next one. Notice when you're doing these dihybrid crosses on your own, you need to make sure that you have 2 alleles instead of just 1. And ironically, look, they're all the same. So in this particular case, because you have 2 homozygous parents, the offspring are all going to be heterozygous for both traits. So the genotypic ratio, they're 100% big T, big Y, little y, and 100% tall stems with yellow seeds. So what if we take 2 of those F1 generation plants? So here, let me go back to this. This is the P generation, the parents. F is the first filial generation. So we're going to take, because it's plants, it doesn't matter, 2 of these, 2 heterozygous individuals, and cross them. So the genotype for each of those plants is heterozygous for both traits. Now, how do we get their gametes from that? There are going to be 4 gametes. Parent 1, let's do the FOIL method. Firsts, big T, big Y. Think of all the possible combinations you can get of those 4 letters. The outers, big T, little y. The inners, little t, big Y. And the lasts, little t, little y. Notice all 4 of them are different in this case. Parent number 2 has the exact same genotype, so they're going to get the exact same gametes. Okay? So let's take these gametes and put them on the Punnett square. So it's going to look like this. Now we have a lot of options. So the first one, if this sperm fertilizes this egg, you're going to put the alleles back together, big T, big T, big Y, big Y. This sperm fertilizes this egg, big T, big Y, little y. Always putting the dominants first when you reconnect the allele pairs. Now we have big T, big Y, big Y. Big T, big Y, little y. So as you do that for each, because each of these is going to be different, you really won't see a pattern until we go back and look at the ratios. So I will let you fill those in. You can pause this video at any time. And you can see there's a lot of different possibilities. But what's interesting is really you can either be tall and yellow, or you can be tall and green, or you can be short and yellow, or you can be short and green. So even though we have 16 squares here, there's only 4 ways these plants can look. So let's take a look. This one is tall and yellow. This one is tall and yellow. This one is tall and yellow, and this one is tall and yellow. In addition, tall and yellow, tall and yellow, tall and yellow, tall and yellow, tall and yellow. And then what if we have tall and green? Well, here's tall and green right here, tall and green here, tall and green down here. What about short and yellow? Well, here's short and yellow here. Here's short and yellow. Here's short and yellow. And only one is going to be short and green. So the genotypic ratio, you just count how many different genotypes you have. It's crazy. Put it all together, it's a 1 to 2 to 2 to 4 to 2 to 2 to 1 to 1 to 1 ratio. On the other hand, the phenotypic ratio, much simpler. 9 tall with yellow seeds, 3 short with yellow seeds, 3 tall with green seeds, and 1 short with green seeds. So all you have to do, and what I can't show you on the same screen, is you look at that giant 16 square Punnett square and figure out the ratios. That's how that works. You also have the videos that you can reference. And that's a 9 to 3 to 3 to 1 ratio. Neither one of these can be put in any lower terms because they have a 1. So there's another picture of a dihybrid cross and how to break down the ratios. In AP Biology, we figure out tetrahybrid crosses. What if you look at 4 traits at the same time? That's quite a Punnett square. So you have on your exit ticket the dihybrid cross to complete. You can watch the video clips of it, and then you have some extra problems. So that's what I have for you today on dihybrid crosses. So you can fill out that worksheet. You have a little bit more to that worksheet. And then you have some practice, okay? There's a practice, and here's more dihybrid cross practice in your packet that you already have. If you're interested in doing those, feel free to do so. I'd love to see if you decide to do that. And you can send that to me, take some pictures and email it to me to see how you do there with that. So any questions, don't hesitate to email me, and have a great day.